Thank you very much, Koei, and I'm very pleased today. I've been on the webinar series as the co-PI of dkNET, as Koei indicated, but I'm very happy to be able to present another project to you that our lab is involved with called the SPARC project, which stands for Stimulating the Peripheral Activity to Relieve Conditions. And the subtitle of my talk is Advancing Bioelectronic Medicine Through Open Science. So the structure of my talk is going to follow this. First, I'll introduce SPARC and the field of bioelectronic medicine, give a high-level overview of what SPARC offers, and then spend some time, since this is really a tool-focused webinar series, about the current SPARC portal and what you can do through it. And then I want to finish with a unique resource that my group in particular has been developing through SPARC called the SPARC Connectivity Knowledge Base, or SCAN. And then a brief consideration of where the SPARC project is going. Koei already mentioned that I am a founder and I have equity interest in Inc., a tech startup company providing services in support of enhancing transparency and reproducibility in the scientific literature. So first, what is bioelectronic medicine? This was a term that I learned when I joined SPARC. It's given rise to a whole new field called electroceuticals. It's also called bioelectrics, neuromodulation therapy. But basically, through the US Brain Initiative and also all sorts of additional projects at the convergence of medicine, neuroscience, and engineering and computational science, there has been a large increase in the use of devices such as stimulators, transmagnetic activation that help both diagnose and treat diseases. So therapies, major companies, Medtronic is one that's probably known to most, have been developing therapies for heart failure, sleep apnea, hypertension, PTSD, stroke, diabetes, Parkinson's. You can see that it is really not restricted to neuroscience, but is really dealing with a whole host of conditions that affect humans. And like with a lot of translational science and clinical trials, there have been a lot of failures here. There's a lot of hype around this and a lot of failures as well, even as some of these therapies have really shown to have a significant impact on quality of life and in treating these conditions. So because of this emerging field, and because there's been a mixed bag of responses from the extremely powerful to the failed trial, the SPARC project was initiated by the NIH in order to address the challenge that we have limited understanding of which neural targets are being influenced and how to precisely control organ function. So people are putting electrodes in, they're stimulating them, they're getting results, but we really don't know very much about the underlying biology. And because of that, we can't do things like try to minimize off-target effects and other sorts of things that you might imagine would happen when you are just putting electrodes in and doing wide field stimulation. So the opportunity that's presented themselves, if we do know this, is that we would be able to develop more effective and targeted neuromodulation therapies by putting neuromodulation or bioelectronic medicine on a much firmer scientific and technological grounding, a much firmer basis. So the idea of the SPARC project, the goal of the SPARC projects, is that we are delivering detailed, integrated, functional, and anatomical maps, data, and models of the autonomic nervous system in particular, and its innervation of target organs. So there's a lot of basic science involved in SPARC, but really its goal is for clinical medicine. So how is this being done? Well, there is a large consortium that has been supported by the NIH Common Fund. So these are big trans-NIH type projects. Currently, there are well over 60 research groups spanning 90 different institutions and companies working across over 15 organs and organ systems in 8 different species in order to provide the scientific foundation that I was referring to in the previous slides. Currently, we have over 150 curated data sets and computational studies that are available through SPARC, and this is constantly growing. And the group that has been charged with coordinating and fielding the infrastructure for SPARC is something called the SPARC Data and Resource Center, or DRC. And I'll be referring to that a lot during my talk because my group is in fact part of the DRC. So Here's just a little bit about how the DRC is organized. It's organized around 4 different cores, and you can see that they are computation and simulation, or SIM core as we call it, data coordination, which is also referred to as DAT core, map synthesis. This is largely the purview of the Auckland Institute of Bioengineering, or ABI, and this is also referred to as map core. And then knowledge curation, knowledge management and curation, which is what my group is primarily involved with. We also refer to it as K-core. It's not that you need to remember the names of the cores, but sometimes they sneak into my slides. I also will acknowledge Gazelle. Gazelle is actually a company that deals with user experience and user interface. So SPARC has tried really hard to make sure that the tools and data that are made available are done so in a way where they can be maximally taken advantage of by the scientific community. So, oops. What does SPARC offer? Well, at a very high level, we offer public access to FAIR data. So FAIR, of course, stands for Findable, Accessible, Interoperable, and Reusable. If you've been attending dkNET webinars, you already know that term, that acronym extremely well. And these are very diverse and large experimental and computational data sets. SPARC has also invested a lot in data curation, which a lot of projects don't. So these are very well curated. They're curated to common standards. And if you kind of look at the titles of these, you see that they're very, very diverse, from experimental mapping data sets to safety testing of predicate devices for fecobionics, which I've learned is a term, along with computational data sets that are seeking to sort of understand how these organs and nerves interact with each other. So it is a very, very rich and unique set of data. And I will spend some time in the next section really going through how you can explore this. But SPARC is not just the data, it also provides a platform for computational modeling and analysis. This is what we refer to as the OSPARC platform, and it is a product of SimCore. And this is a very powerful simulation environment where we can take data visualizations and things from SPARC and do computational modeling and simulations on them. So here you're seeing autonomic neuromodulation of organ function in one of the things that are made available. Okay, these are open resources and online. They are sophisticated and do require a learning curve, but these are also available through SPARC. We are also mapping our data to common reference spaces. So as I'll show you later, SPARC has produced a library of computational scaffolds. These are realistic models of species-specific organs that are available. So you can use that to map data on it. For example, here you see distributions of cells in the context of the anatomy of the heart. But they can also, again, be used as the basis for doing those types of simulations that you saw, realistic, spatially realistic simulations using the OSPARC platform. But we are really using this to make sure that all data that comes out of the heart is mapped into a common reference space so that one can easily compare across species and also across data sets. Finally, SPARC, as I mentioned in the very beginning, with its entire purpose, is building maps and knowledge bases of ANS circuitry. So I'll show you a little bit later the map interface. You can see the wiring diagram that is representing connections of the major autonomic nerves. This is all being collected into a knowledge base, a computable knowledge base called SCAN, and I'll be presenting the details of that in an upcoming part of the lecture. But this is a very, very powerful product because we are really going down to very granular levels of how these nerves exit or enter the central nervous system and make their way through individual nerves and nerve bundles to interact with end organs. So let's take a look inside the SPARC portal and see how you can use it now. So the SPARC portal is the gateway to SPARC. In fact, SPARC is made up of several independent platforms, as you saw in the overview of the DRC. But the SPARC portal provides the uniform skin and services over those 4 platforms so that you can use it effectively. As I'm going to refer to, this is supported on the back end by something called the SPARC Knowledge Graph, which is a building connections between data, metadata, knowledge, and tools to make it easy for you to be able to navigate across the rich set of resources that SPARC provides. This is a work in progress. We're constantly adding data and constantly adding functionality. And if you'd like to see how SPARC is going to develop, you can actually access the SPARC roadmap, which is available online. As I mentioned, even though this is difficult and slow work, we are really trying to invest in the user experience and the user interface to make it as easy as possible to use. So there's extensive user testing that goes on with outside users to help us develop the user interface. So to get started with SPARC, if you look at the home page, you can see the top navigation tells you that you can find data, tools and resources, maps, news and events, and about. There's several ways that one gets started. Usually people start by looking at the data we have. You can look at all the data by clicking on Find Data, or you can see down below, we have these little organ icons where you can click on them and be immediately taken to the type of data that's associated with that particular organ. There's a help section over here if you get lost, and if you're interested in global search, you can search across the entire portal using this. So there's many ways to get into it. We're going to start by, for example, searching on the colon icon by clicking on the colon icon. When you do that, you are taken to a list of data sets that are associated with the colon. As I mentioned, these are all curated to the same standard. So you can see here that we have anatomical structures and species and so on and so forth. There's additional search functions that are available. I'm going to focus on data now, but I will talk about how you can interact with models and simulations in just a bit. But essentially, SPARC has launched a new faceted search capability that makes it easy for you to access these and explore the knowledge graph. So over here, you can see some of the facets that are available to you. You can click on anatomical structures, species, experimental approaches, and other things to make it very easy once you're in the SPARC portal to navigate across them. The SPARC knowledge graph I mentioned before means that once you're exploring a data set, it becomes very easy for you to then go and key off these different facets in order to be able to search it further. So for example, here is a colon data set on the human colon. And if you're interested in more human data, you can go ahead and click on that, and it will take you to the list of data that are available for human. You can again, filter this or explore it more by technique, species, sex, age, authors, and constantly new facets will be released because SPARC is built on a very, very rich set of metadata. It's just a process of figuring out how the best way to expose this information is without overwhelming people. But I think you can see it's a pretty nice interface. So let's look a little bit into the anatomy of a SPARC data set itself. One of the things that's nice about SPARC is that it is curated. We have a team of curators. I see some of them on this webinar that make sure that all SPARC data sets undergo quality checks and that they are aligned to a set of uniform standards, right? This, of course, is the heart of FAIR, is that these things are aligned according to a uniform standard. So if you click on the files menu that is associated, there's a tabbed interface that comes with each data set. And if you click on it, you will be returned a list of files. And I don't want to go too far into it, but every single data set that you see will be organized in the same way. So if you understand how to navigate one data set, you can navigate them all. And I think you can agree that that's a very powerful feature because SPARC is dealing with all different types of data from microscopy, physiology, genomics, devices, computational data, and making sure that your experience when you go and navigate through these is similar is a very, very powerful feature. You can download these data sets. Some of them are very large, so not all of them can be downloaded directly from the web interface, but if they can't, there's a way of downloading them via Amazon Web Services. And you can also, if you don't want the entire data set, download individual files for your use. The nice thing about SPARC 2 is that there's a rich set of metadata that is collected for each one. So you see that there's notes about how the data are organized, what was the point of the studies, additional curators notes that tell you about experimental design, subjects, and samples. But one of the things that are required of all SPARC data is that they be linked to a detailed experimental protocol so that you understand exactly how these different data sets were acquired. These are being stored in protocols.io, and the links are provided on the landing page of the data set. So if you click on that, you see that it provides you very detailed experimental data, which is another feature I think that distinguishes SPARC from a lot of projects, is that we do provide this very rich context to maximize your ability to be able to use these data sets. SPARC is also, the data sets are linked to online viewers and exploration tools. These are constantly being developed and released, but if you go to a SPARC data set and you click on the gallery, you will be taken to a set of viewers. For example, you can view movies online. There's a lot of movies that are spectacular of these very, very large, beautiful data sets. You can also explore individual images, even large images, using some of the online viewers. So even though you can completely access the data by downloading it, we're trying to build as much functionality into the portal as possible so that you can do online exploration as well. One of the things about SPARC that I mentioned, and I'll get back to in the second half of the talk, is that we are building connectivity maps and connectivity knowledge bases. So one of the things, if you actually explore a SPARC data set and go to the gallery, that you will often see is something called a flat map. If you click on that flat map icon, it actually tells you whether SPARC has information in the knowledge base about connectivity of the target organ. So this, for example, is a data set about bladder. And if you click on it, you can see that there's a rich set of connections that are coming from these connectivity maps that I showed earlier that refer to the bladder. Now, I'll talk a little bit about how the connectivity map is being developed. And so you don't always get this level of detail, but you can see that SPARC is allowing you to place data sets and data experimental results in the context of the connectivity of the autonomic nervous system. I want to spend a little time about models and simulations. Our group works on the data side and connectivity side, so that's what the bulk of my talk is going to be about. But I do want to remind you that we are linking a lot of the data sets and models that are available on SPARC to the OSPARC simulation platform. So if you go under the models and simulations section over here, it will give you a list of computationally derived data sets. These are not experimentally derived, but they are computationally derived. And if you click on some of them, for example, this one here, Jupyter Notebook Exploration of Computational Tools, you will get a button that says run simulation. If there's a run simulation, it means it will take you to OSPARC, the OSPARC platform, where it will actually allow you to execute and run the model that is presented in SPARC. So in this way, we are linking across the data sets that are in the SPARC portal and the computational and modeling tools that are available. And I think you can see why SPARC is such a powerful platform. In this case, users can use and publish things like Jupyter Notebooks, which allow you to investigate a specific aspect of autonomic function. This particular Jupyter Notebook deals with the excitation properties of computational models of unmyelinated peripheral axons. This was a computational data set that was published by one of the SPARC investigators, and they also made available a Jupyter Notebook that lets you actually run some of these routines on the OSPARC platform. I mentioned these 3D scaffolds that are also being produced. These are being used as a common coordinate system, but they're also available to researchers to use in whichever way they want, whether they want to build a simulation on top of them or they want to use it for visualization. So if you go to SPARC and select a scaffold in that in the modeling and simulation section, you'll be presented with a very colorful library of three-dimensional scaffolds. These are species specific, so you'll notice that this is the colon in the pig. I think this is the colon in the mouse over here, but these are all available for you to use in your research. And again, how are people using these? Well, these are being used for whatever purpose you want, but also we are mapping data that is coming out of those SPARC data sets into these 3D scaffolds so that we can compare across them. This is just one of the papers that was published where they were looking at the distribution of intrinsic cardiac neurons across the sexes in, I think it was in the rat, and showing variability of it. So these data sets, these mapped data sets, will also be made available through the gallery as they become available. Okay. So that was just a brief introduction to the SPARC portal and how you can get started with it. Now I wanted to spend some time on the sort of what I consider the heart of SPARC, which is the knowledge that we can extract about ANS circuitry and how it interacts with end organs. And I already alluded to the fact that we have a knowledge base called SCAN that we are building here. So we'll talk a little bit about how that is being built and how it is being populated. If you go to the SPARC homepage, you will see a section that is called Maps. I showed you that from the SPARC data set, you can get to the maps. Well, it turns out that you can also go to the maps and get to the SPARC data set. So if you open Maps, you will see a series of species-specific maps: cat, pig, human, mouse, and rat. And if you click on one of them, this is the rat one right here, it will show you some cabling which represent the different types of nerves. They are color-coded according to standard nomenclature: parasympathetic, sympathetic, preganglionic, and postganglionic. But you'll also see these little buttons right here, these little icons. If you click on those, they will actually open up data sets that are associated with it, models and simulations that are associated with it. So you can use the map interface to get back to the data, just as you can use the data to get to the map interface. And that's what I mean, that it is a highly integrated portal that's building across the three platforms. So it's very, very powerful, and there are multiple ways in which you can explore it. If you zoom up on this map, I'd say that the rat map is the most heavily populated. You can see that the level of circuitry is actually quite detailed. So it is not just simple organ to organ, but it does, in fact, map the level of circuitry at a sub-organ level, and also allows you to look at a little bit of the circuit diagrams that are associated with each of these. So if you zoom up on it, it reveals additional detail so that you can explore this in more depth. So I wanted to spend a little time on how SPARC is building this knowledge base and what is going into it. So the ANS Connectivity Map is currently being populated on a system-by-system basis. So you may have noticed when you look at our maps, there's nothing for the liver, there's nothing for the kidney. And these are being gone through and systematically being mapped in terms of circuitry, but that takes some time. So as these models are developed, they are released. So we're doing it on a system-by-system basis. I showed you the level of detail that you can see, and we already saw, I think, this diagram right here. What we call these are these detailed subway maps of connectivity that are being built through consultation with anatomical and physiological experts in SPARC, consultation with the literature, SPARC data, and also something called the Anatomy Working Group, which has external anatomists who vet this information to try to put it in. So if you look at these maps, they are very, very detailed. They not only have the locations of the soma and the locations of the terminals, but they plot the routes by which these exit or enter, which vertebrae they go through, which structures they hit, what nerve bundles they go through. So these are very, very detailed topological maps of connectivity. And if you compare those to the sort of simple maps you get in Gray's Anatomy, you can see that this level of detail is really quite exquisite. Once we have these maps in the current SPARC interface, these are manually drawn onto these diagrams over here. So there's these maps are produced, they're SVG images, they're all available if you'd like to see them, and then they are manually drawn onto the map that you see through the SPARC interface. If you look at that in a little bit detail on the map interface, you can see again that there are ganglion there are little cuffs which represent the nerves, and then you have the individual neuron populations that are coming from these models coursing through the nerves. So this is the level at which on the flat map, the connectivity of the autonomic nervous system is represented. Currently, in SPARC, 6 app anatomy models that are available. So we have one of defensive breathing by Steve Lewis and Don Bolser, Janet Keith's bladder model, bronchomotor control, model of the heart. You saw that detailed information on the heart, model of a distant distal colon, and Terry Powell's model of stomach innervation. As I mentioned, these are all SVG images. They are all available for download if you would like to see and explore them. Very stylized, they're highly stylized, but I think very, very interpretable. But I think you can see that there are limitations to the current approach of taking these very detailed maps, creating manual drawings of them, and then transferring those drawings onto the maps that are available through SPARC. First of all, it's very difficult to keep pace with new connectivity knowledge, very difficult to correct errors. There are always errors in something that is this complicated. The information here represents very granular connections and routes. That's very hard to display on an interface like this. So you're only getting a subset of the information that is available on these maps onto the current interface. It's very difficult to understand connectivity patterns, looking at these very complicated diagrams, following where they go. Comparing across species is difficult. And even though we're having people, experts and create the maps and vet these maps, there are still points of contention. There are still things which people disagree about, and it's very difficult to sort of represent this in this manual context. So because of this, we have been working with the Auckland group, who's developing the map interface, on developing a knowledge base for autonomic connectivity. What that means is that we can take the information that is being produced in these diagrams and we can turn that into machine computable information. We are doing that using a tool called App Anatomy, which I should have explained a little bit earlier. App Anatomy is basically a model for being able to take these very complicated things and turn them into something that a machine can process. These all go into a graph database called SciGraph, and involves, again, experts coming together to define them, translation of these into a computable language by knowledge engineers, and storing in SciGraph so that they become available for query. And I want to acknowledge the work in our group by Tom Gillespie, Monique Sorla-Ziegler, and Bernard De Bono, who are really leading the way on this. So here's just a little bit more detailed on App Anatomy. Basically, it is a modeling system or a platform for modeling flow between biological structures, and flow can be fluid, it can be information, it can be whatever type of quantity is transferred from one place to the next. But the important thing to know about App Anatomy is that it represents neuron populations and the locations of the individual parts at very fine detail. So it represents the locations of the somas, however many axon segments there are that traverse all of these different places, the axon terminals, the layers in which these axon terminals are found. It is a very, very granular model of neurons and their parts. And App Anatomy is really responsible for taking all these individual parts that comprise a neuron and putting them back together in a way that they can be computed on without losing all of this detailed topological information. So what can we do with information like this, which is in SCAN? Well, in fact, it provides a very powerful substrate to be able to support knowledge-based queries. So you can you can find anatomical regions that are traversed by individual neuron types. You can find connections that course through a specified tract nerve or ganglion. One of the other cool things that you can do, coming back to the maps that are being produced here by the Auckland group, is instead of drawing all these lines by hand, you can now automatically populate these models. So this vastly simplifies the ability to be able to produce, keep up to date these types of maps, because now they're being driven by queries into a central knowledge base rather than being drawn by hand. And you can also provide detailed provenance of each of these connections. So every one of those statements about where these things traverse usually is attached to a body of literature, a data set, something else. And it becomes very easy for you to find the information that goes into the producing of these maps so that you can validate and whether you find it trustworthy or not. But that also means that if there's a controversy or something else, you can include annotations on top of this that make it easy for you to understand the state of knowledge. So I want to go through in the next section a few of these use cases so that you can see how we're using SCAN inside of SPARC. So first, some of those knowledge-based queries, right? If you look at this diagram here, you can see that there's these neuron populations. They have very, very complicated roots. But what SCAN allows you to do is issue a query that essentially lays out all of these structures. And this is what the result of the query looks like, which is a little bit complicated. But over here, you see it in a schematic diagram. These are identifiers that come from one of our ontologies that are used by SCAN, so you can ignore them. But essentially, it tells you that these two neurons, these are all the structures that it traverses, including the different nerves over which they traverse, and then the things that they target, in this case, the pelvic ganglion. So you can see the number of individual anatomical structures that are impacted by this one neuron population, but this was all retrieved and visualized using query rather than having to do that integration yourself using these SVG files. So because we have this, as I mentioned, we're now moving towards automatic visualization of connections on the SPARC flat map. And what you can see here in the middle, this looks like the flat map that you've seen with the rad, but in this case, it has a neuron that is drawn. This was completely drawn through a query to the SCAN knowledge base. That's why there's little gaps in here where the rendering has maybe not been closed completely. But I think you can see that this is really an amazing step forward. So all of this routing was produced by an automated query into SCAN, which gave the locations of somas, axon terminals, and then also all of the anatomical structures over which this traverses. Now, as I mentioned, it may not represent all of that, but it is able to represent at the level of granularity represented by this flat map, the information that is in SCAN. But if you were to launch a query from this, you would be able to get that detailed topographical information. So I think, again, you can see that this is a very major step forward, and this will be released soon to the SPARC community via the portal. So that makes it easy, as I mentioned, to be able to attach provenance. So now if you go over one of these neuron populations, it can take you to the references that were used to create these maps, gives you a very rich source of information. It connects you to data sets and other things that may have information relevant to a particular connection. All of this will be made available as the new interface comes forward. But it also lets us do some really interesting integrative biology as well. So I mentioned that we had 6 maps, I think this only has 5 of them, models inside of App Anatomy. One of the things we're doing is making sure that when we put these things into SciGraph, we're using the FAIR data principles. So even though these are separate models, you might see figures like this in a set of papers, and they're all separate. They reference common anatomical structures, and all those anatomical structures, as you saw in that one slide that had those Uberon identifiers, are using common identifiers. That means no matter what model a structure appears in, we can use that as a point of connection in the knowledge graph. So you can ask a question then that says, find all neurons with processes in the inferior mesenteric ganglion. And it will look across all models, not within a single model, but all models for anything that is found inside the IMG. And what you're seeing here is a list of those neuron populations that are coming through. Interestingly, these neuron populations are coming across from 2 different models. So there's information that's coming from the colon, and there's information that is coming from the bladder as well. But inside of SCAN, this is all available for query so that you're not getting some of the connections over here in the bladder and some of the connections over here in the colon. Because we have this level of integration, it means that we can break down this process of population into a series of discrete steps. That's why we're producing the maps on a system by system basis, but get the knowledge base into a unified structure so that we can cross query. So you can see, I hope, that this is really laying the foundation for one of the things that SPARC is trying to achieve, that you would be able to do virtual in silico medicine by putting a stimulating electrode or a recording electrode, whatever you want, into a given location. In this case, it's the inferior mesenteric ganglion. And you would know what neuron populations are sending processes through these. You can even go down a level because of the detail that's available in app anatomy and understand that you're seeing segments of axons, segments of dendrites, somas that might be in the ganglion itself, right? You are actually influencing all of these if you stick a stimulating electrode in there. So I just, as a sort of little virtual experiment, I thought it would be interesting to say, if we actually look at all the anatomical structures that are impacted by the neuron populations that we have in the knowledge base, and we don't have all of them yet, because as I mentioned, we're doing this on a system by system basis. You see that by putting this electrode here, in fact, you hit over 10 different neuron populations that traverse over 30 anatomical compartments. So even at this little, at this initial level of population, you can start to see how the scan underlying the tools of SPARC is a really powerful addition, because it is going to let us, to the best of our knowledge, understand what is being impacted by a perturbation in any region of the autonomic nervous system. Okay, so I think we're coming to the end here. SCAN is also allows us to provide a very flexible knowledge visualization environment, which I think is really, really important. And we think that databases like SCAN really will provide the basis for next generation of atlases. Imagine instead of having paper atlases, you have a computational atlas, which is driven off this knowledge base, though you can visualize it whichever way you want, interact with it, query it even further. But you don't just need to produce these flat maps. In fact, you can visualize this information in multiple different ways. All of these types of tools are being used inside of SCAN, depending on what it is that are inside of SPARC, depending on the particular use case, wiring diagrams, 3D models, atlases. All of those are being driven off of a common knowledge base. Okay, so what's next for SPARC? Well, looking forward, the SPARC portal and tools will continue to increase in breadth, depth, and functionality. All of the things that I showed you in the early part of the talk are all available through SPARC. They're not always easy to find. For example, if you want to find data sets that are associated with simulations, you kind of have to hunt a bit, but we're constantly improving that. We're constantly taking the information in the knowledge graph and exposing it on the user interface. And so the SPARC portal will continue to evolve over time. Really, one of the directions that we're focusing on is to make it easier for you not only to explore inside the portal, but to make those leaps over into OSPARC or other platforms where you might want to take SPARC data and use that as the basis of a computation. But SPARC is also going to continue to develop as a community hub for ANS research. SPARC is the place for autonomic research. It's the largest database and knowledge base of information available on that. So we're building in features that will provide collaboration spaces for not just SPARC researchers, but we're expanding this out to the broader community. So you do not necessarily need to be part of SPARC in order to make use of these things. And we want to broaden participation beyond the SPARC funded consortium, but also start linking, for example, to other projects that are producing information that might be useful. So SPARC is in the 5th year of a multi-year project, and I'll talk a little bit about phase 2 in just a moment, but the SPARC project will continue, and there's many opportunities for people to participate. So one of the things that we're working on with other projects that are funded by the NIH Common Fund. NIH Common Fund is producing some very, very large, well-populated data resources. You may have heard of HubMap, which is spatial organ gene expression data sets, reprotox on teratogenicity and others. So there's a whole many different projects that the Common Fund is funding, and they're really putting an effort into taking FAIR seriously, which basically says, right, these should all be interoperable. So SPARC is participating in this CFDE ecosystem, which is fielding a portal. You can see the URL down here that will let you cross query across all of these assets. And just like we are using ontologies to unify the anatomy references inside of SPARC, all of these projects have agreed that they're going to be mapping to these common ontologies. So that should make it very easy once inside of SPARC to be able to go across the different assets that are being assembled by the Common Fund data ecosystem. But we've also really emphasized in the CFDE that SCAN is a really unique asset. We don't know of any other database of connectivity, especially of the autonomic nervous system, that has the breadth of what SCAN does. And I think if you start to, again, think about bioelectronic medicine, SPARC is doing a lot to map against individual, to create data sets and to create this knowledge base. But it would be great, for example, to be able to use connectivity information contained in SCAN to interpret gene expression patterns that you see across organs. And so some of these different partnerships that we're involved in is how we can take this unique asset and use it as an overlay on top of these other large mapping projects, because obviously coordination of gene expression and many other things across organs is going to be done at the level of nerve connections as well as other types of fluid flows. So as I mentioned, SPARC is also a community hub. So if you go to SPARC, the SPARC portal, it will tell you about new initiatives that are coming out. SPARC is about to go into a new phase. Instead of broad mapping across all of these different nerves and organs, it's really focusing on the vagus nerve in part 2. So there are various RFAs out that you can access through SPARC to try to find out about these. And I also want to just give a shout out to the SPARC X Prize competition. So NIH has launched a new prize called the Neuromod Prize to incentivize selective neuromodulation of multiple outcomes without off-target effects. And you can also find out about that on the SPARC homepage. The submissions are due on April 1st. I think it's $800,000 or something like that in prizes. So it's very, very significant. And with that, I just want to thank you for your attention. And I also want to thank all of my colleagues in SPARC, especially KCOR, Tom, Monique, Joe, Barak, Bernard, Natalia, but also the curation team and others who are involved in making SPARC what it is, and my colleagues at MapCore and all of SimCore and DATCore as well. So thank you.
Thank you, Maryann. And so now it's our Q&A session. People, you can unmute yourself to ask question or add your question using the chat function. So during the presentation, there is one question. I think it's probably in the very beginning of the presentation. How to find a connectivity map from dataset? Can you show us again?
Oh, sure. Oops, let me go back. Oh boy, it's taking its time. There we go. Okay, so if you are in any data set and you click on the gallery, okay, so if you click on the gallery, inside the gallery, it will give you various visualization options, and one of them will be called flat map. And right now, it's not very user-friendly because it just gives you an Uberon identifier, but essentially it's taking the organs that are represented in this data set and then taking you to the part of the connectivity map that is associated with that organ. So in this case, it's taking you to the connectivity map of the bladder. So it should be very easy to get there. And then once there, you can zoom in and out. It does not have, this viewer, have all the functionality that you see by accessing the maps from that top level navigation, but at least you can explore the connectivity map that you see by zooming and panning and so on and so forth. Does that answer the question, I hope?
And there is another question.
Is it possible to have a recording of the presentation? Yes, I think Koei will give you details about that. So all of these presentations are recorded and made available through the SPARC website. I'm also going to give Koei a copy of my slides because my slides have a lot of hyperlinks and things in there that are often very useful. And that way you can have that as well.
There's a question from the chat. Is SPARC open for community to submit tools and the data set? Do I have to publish it first before submit?
No, you don't. That is a very good question. So yes, SPARC is right now, we are in a transition phase in SPARC. So if you actually look, and I guess I could just look and do this live. It's always dangerous, but it seems like it's working. Okay, so if you look under tools and resources, if you actually build a tool that works with SPARC, you'll see that we have lists of things here. Some of them come out of SPARC. So I mentioned app anatomy, for example, but a lot of them don't, right? A lot of them are just associated tools that are useful in the context of SPARC. So if you were to build a tool that works with SPARC data, for example, we would be very happy to list that here. In terms of putting data in, we are in a transition phase where SPARC was initially populated using people who were funded by the SPARC consortium. That is, NIH said, you are getting money from this consortium and you will put your data into the SPARC repository. But SPARC is meant to be an open repository. And so we are looking for people who might be interested in putting their data in so we can work with them to see what they're interested in doing, how they're interested in putting it in. You do not have to have your data published in an article. We consider publishing data here to be publishing your data. So if you look at, I didn't go into this in great detail, but each one of these data sets is given a DOI, for example. So each one of these is considered published. It has a DOI. It has full citation metadata so that if somebody uses your data set, they can put this citation into their paper so that you can get credit and people also know what data is being used. So we hope that if people are interested in actually using SPARC, that they will contact us, and we are anticipating that by the end of the year, it will be open for data that is coming from outside of the community.